Genetically modifying pigs for organs is a new and controversial topic to society. My name is Alexa McLong, and in this video I will be going over xenotransplantation, the history, the modern research, risks and complications, and ethical concerns. To start off, organ transplants are not common, with the majority waiting their whole lives on the wait list. An average of 10 patients will die each day from the list consisting of around 48,000. Organ donations are rising but not quick enough. Therefore, biotechnology has introduced a new procedure called xenotransplantation, which is the process of transplanting cells, tissues, and organs from one species to another. With the use of xenotransplants, patients could have their lives extended or aid in their daily lives. The appropriate audience for this operation includes people in need of a kidney, heart, lung, liver, pancreas transplants, skin grafts, corneal transplants, limb and bone transplants, burn survivors, as well as those with Parkinson's, diabetes, and Alzheimer's. The study and experimentation of xenotransplantation has existed for decades, but started to spark curiosity in the 1960s. Primates were the primary focus for what was viewed as the most qualified donor. During the early experiments, chimpanzee kidneys were transplanted into humans, with one subject having their life prolonged an extra 9 months. From 1963 to 1993, 31 clinical procedures involving transplantation of solid organs were performed in the United States and South Africa. The most publicized operation involved a child named Baby Faye who was born with a malformed heart. The infant survived another 20 days with the transplanted heart from a baboon. Presently, the main animal of choice is the domestic pig because the anatomy is like humans, pig breeding is well developed and cost effective, and the large variety of breeds allows the size of the organ harvested to be matched to the patient. It seems very straightforward, but the species difference presents a range of immune complications after the transplant, which can lead to non-functioning organs. The immune system job is to attack unknown bacteria and keep it out, and as expected, it does just that to the pig organs. Most threats that could be harmful to humans can be eliminated by careful donor selection, breeding in sterile and isolated conditions, and screening of each donor candidate. Porcine endogenous retroviruses, also known as PERVs, remain a problem as they constitute an integral part of the porcine genome. As seen in this photo, PERVs are inactive in pigs, but when implanted, can be activated and infect the recipient's body. With the precise adjustments, pigs have the potential for being a qualified donor by using different ways of altering the cell, which could be viral transductions of DNA or modifications of the CRISPR bacterial immune system. To start the process, the double-stranded helix of DNA needs to be broken in order to change the original nucleotide sequence to create a mutation in protein synthesis. When the DNA is broken, the engineers can make the changes in the amino acids. With all this new technology, there are people that have their concerns. Some believe that the process is unethical and going against the creator of God by creating creatures that would have never existed. Other worries are unknown risks that could threaten the public. The research is not complete yet, and carelessly carrying out operations could cause trouble. In the end, Xeno has the potential to save thousands of lives in the long run, one patient at a time. The new science is still going through complications and concerns, but with the proper technology, the future of xenotransplantation could be closer than we think.